Perfect, thank you. Alrighty, thank you everyone. Sorry we're a few minutes late, but thanks for sticking around. Today we have Alec Piper. He is a master's student here at the University of Nebraska, working in the Center for Advanced Land Management Information Technologies, CALMIT, under Drs. John Gammon and Dr. Sabrina Russo. He earned his bachelor's degree in biological sciences from Doane University in December 2020. Alec's master's research focuses on the variation in photoprotective pigment responses among trees using proximal remote sensing. Studies investigating variation in species photoprotective strategies aimed at better understanding the mechanisms regulating photosynthesis and photoprotection can enhance the ability to monitor plant stress in forest communities with remote sensing technology. So we'll go ahead and turn it over to Alec if you want to tell us all about it.
Cool, thank you. Do we want to dim the lights in the front, or can everybody? Yeah. Okay, cool.
Okay. Yeah, that works.
Okay, cool. Thank you for the introduction, Bailey. I'm Alec Piper. I know a lot of you in here, and some of you joined me during my dissertation or thesis defense. So this is kind of the same research that I'll be presenting as I did for my defense. So my project is looking at assessing photoprotective pigment responses among deciduous and evergreen tree canopies with leaf spectroscopy. So a little background information, plant photosynthesis is a critical component in ecosystem health. It is the underlying mechanism involved in regulating a variety of ecosystem processes like the carbon cycle, for example, here in this figure. It's also an important component in maintaining habitats for all biodiversity within that ecosystem, as well as ecosystem resilience in response to environmental change. And just as an example of maintaining ecosystem health, it's kind of the gateway or the foundation to the transfer of energy across trophic levels and establishes food webs. So it's very, very important aspect to ecosystems and ecosystem function. And a part of plant photosynthesis, functional diversity is another critical component regulating plant photosynthesis. Here's a figure kind of showing how species diversity increases plant biomass productivity. So it's a critical aspect in regulating ecosystem function and also the stability of ecosystems. And functional diversity describes the variation or the differences among species and how they utilize and acquire resources, and those differences are often expressed in variation or differences in plant photosynthesis. But productivity is largely dependent on conditions favorable for photosynthesis. So in response to environmental change or environmental conditions that are unfavorable for plant photosynthesis that limit photosynthesis, plants can adjust their light use efficiency. So how much of that light is actually realized and fixed as carbon via the xanthophyll cycle or by altering xanthophyll pigment pool sizes. So these are photoprotective pigments. When environmental conditions are constraining the amount of photosynthesis that can occur. Here's kind of a figure showing we have net photosynthesis on the y-axis and light intensity as an example of a limiting environmental condition affecting or constraining photosynthesis. So here's kind of our linear response if environmental conditions were favorable, but as we get changes in environmental conditions that are constraining photosynthesis, we see that that kind of tapers off, and that's called an adjustment or changes in light use efficiency. And the xanthophyll cycle, these represent different mechanisms of photoprotection. I'll get more into this, but really briefly, the xanthophyll cycle is a fast photoprotective mechanism, and xanthophyll pigment pool size changes are kind of a slower photoprotective mechanism, and I'll get more into that here. Now, our ability to detect these are invisible to the naked eye, but remote sensing is kind of the key in understanding and being able to monitor these changes in light use efficiency or productivity within ecosystems across a variety of different species and functional groups or functional types that make up that functional diversity. So with remote sensing instruments, we're able to monitor these dynamic changes in xanthophyll pigments with the Photochemical Reflectance Index. We can measure the interconversion of these xanthophyll pigments in response to environmental change. And then also PRI variation is attributed to those pigment pool size changes. And both of these represent distinct mechanisms. So right here is a schematic of the xanthophyll pigment or xanthophyll cycle, which is that fast photoprotective mechanism. When environmental conditions are changing rapidly, we can see here as light becomes in excess, these are the xanthophyll pigments. Violaxanthin is converted to anthrozanthin into its photoprotective form, which is zeaxanthin, and we get reduced light use efficiency. And that is reversed when conditions become more favorable. Again, these two mechanisms, because they influence PRI, presents kind of a challenge to how we interpret PRI and what mechanisms are associated with those responses. So this can be a difficult when we're trying to understand what PRI is responding to between these two mechanisms and what that means for changes in plant photosynthesis. This is an issue, especially within canopies, as we can get a lot of variation in both the amount of xanthophyll cycle activity that's occurring due to variation in canopy light conditions throughout the kind of canopy gradient. This is kind of showing differences in light exposure. Not only do we get changes in the amount of xanthophyll cycle activity that's occurring, but we also get differences in those pigment pool sizes, and carotenoids represent that slow mechanism or that constitutive pigment pool size of xanthophyll pigments. So how do we characterize this? How do we parse these two different photoprotective mechanisms apart? When we're interpreting PRI, leaf level or proximal remote sensing, we're able to split these two responses. So here is kind of the resting state. Just real quickly to explain how this works. This is assuming this point is the constitutive response associated with pigment pool sizes. And the assumption is that in this resting state, there's no active xanthophyll cycle activity occurring. And we can actually, when we expose this to highlight, expose leaves to highlight, we can track and monitor that fast photoprotective mechanism. And this is the facultative response for that fast photoprotective mechanism. These are termed PRI0 and ΔPRI. Okay, so if I say PRI0 and ΔPRI, PRI0 is that slow photoprotection, and this is the fast photoprotective mechanism. This is really time consuming. It's really difficult to do. You have to dark adapt leaves, which means you either have to bring them and sample them in the lab or sample them before at pre-dawn and at midday to actually get these two distinct responses. But in Gammon et al. 2023 and Wang et al. 2024, they developed this new method to estimate this slope and intercept of representing these two distinct mechanisms using airborne hyperspectral imagery. And based on at the sub-canopy level, based on the variation in light exposure within the canopy, they're able to kind of get an estimate of both of these responses. So this greatly improves our ability to understand photoprotection in the field and also quickly quantify these for a large number of species relatively quickly. Okay, but the issue is, although this is kind of a substantial advancement in PRI and using PRI to measure stress and changes in photosynthesis, there's a lot of things going on in nature, prevailing environmental conditions, the light environments that we had talked about, but also differences among functional groups. And there's a relatively small number of species that have actually been quantified for both of these responses. So there's still a lot of unknowns or uncertainty in our interpretation of PRI and what that means for photoprotection and environmental stress responses in plants. So our study objective and overarching hypothesis. So the goal of the study was to assess variation in how the expression of facultative and constitutive photoprotective mechanisms among tree canopies of deciduous and evergreen species and across the growing season. And our hypothesis is that we hypothesize that the variation in photoprotection is structured by trade-offs associated with leaf lifespan, okay? And we decided to do I chose the deciduous and evergreen comparison because they represent distinct functional types or functional groups, okay? These different species belonging to deciduous and evergreen, they have contrasting leaf habits, which have implications for how they function in an environment, how they utilize resources, and in ecology, they're termed, they have contrasting ecological strategies. So this is kind of a great comparison to assess whether or not photoprotection is structured by or agrees with these kind of ecological strategies that have been defined in a lot of plant ecology literature. And there is some evidence to suggest that photoprotection is phylogenetically conserved amongst functional groups. Cool. So research questions, how do dynamic pigment responses related to photosynthesis and photoprotection vary among tree species having different lifespans, and also seasonally among these groups? So for our first question, we expect that photoprotective pigment responses should be distinct due to their distinct functional strategies for reconciling trade-offs in photosynthesis versus photoprotection. So between these two functional groups, deciduous species have larger or higher photosynthetic rates compared to our evergreens. So we had expected that because they have a large capacity for photosynthesis, they would have a larger dynamic or utilize this fast photoprotective mechanism that complements that higher rate in photosynthesis versus our evergreens that have more constrained photosynthetic rates. So they wouldn't require such a strong, fast photoprotective mechanism. And then for the seasonal variation, these should be associated with their differences in foliar responses to the onset of autumn that are associated with leaf longevity. So deciduous species, they undergo phenology, they grow rapidly during favorable conditions, but evergreens retain their needles. So we expected, because the leaf lifespan is greater in our evergreens, that there would be less variation in both of these PRI components compared to deciduous that undergo leaf senescence. So we should see a larger degree of change in photoprotection because of those differences in phenology. So our study site and species, we sampled here on East Campus. Really, I picked a few species relatively close to Hardin Hall. And I collected data from June to November of the 2023 growing season. And we kind of characterized our seasonal time points from mid-season, June to August, and our late season is from September to November. Here's kind of a table of my species. Red oak, American basswood, American elm, ponderosa pine, red cedar, and white pine are the species. Leaf reflectance sampling, I kind of already went over this, using the dark to light method to obtain these short and fast photoprotective responses. 40 leaves per species were sampled at each time point. Yes, this wasn't included in my research questions, but I just wanted to briefly touch on this sampling because it's related to kind of the research that I'll bring up later and deriving these PRI slope and intercepts using airborne remote sensing. So in addition to that leaf level sampling in the lab using the dark to light method, I collected reflectance values from shade and sun positions as a way of kind of testing whether or not we can actually derive these similar PRI responses that are parallel to the remote sensing imagery studies. Okay, so variation in photoprotection among my species and functional groups. These are the kinetics plots, those PRI-light responses that were derived using the dark to light method. So we can see there's substantial variation first in those differences in kind of how differences among species and how they utilize these slower photoprotective responses versus kind of the rapid, more fast photoprotective mechanism. And we can see here in this subplot between deciduous and evergreens that deciduous species have higher intercepts, but a larger capacity for this fast photoprotective mechanism versus evergreens have a more shallow intercept or a lower intercept and a more shallow kind of fast photoprotective response. Another way of looking at this, okay, PRI-0, remember, is that slow mechanism, okay, that's associated with kind of changes in, or the relative abundance of xanthophyll pigments that are associated with kind of that distinct photoprotective mechanism versus our ΔPRI, which is that fast photoprotective response. So this might be confusing, but lower intercepts actually indicate that they have greater xanthophyll pigment investments, and a higher intercept, higher PRI-0 values indicates that they have less investment in that slow mechanism, okay? And for our ΔPRI, it's just the opposite. This is a slower or a more shallow facultative response or fast photoprotective mechanism, while our deciduous species have kind of a greater capacity in utilizing that mechanism. So when we look at this in kind of principal component space, we can clearly see that there's trade-offs in these photoprotective strategies amongst our different functional groups, kind of illustrating different photoregulatory strategies. Where there's trade-offs, we see that our deciduous species occupy or have a distinct trade-off in their facultative component here, that fast kind of strategy, while our deciduous species favor kind of that slower strategy. And for our seasonal changes, these are again the kinetics plots between mid-season, it's in red, and late season time points in blue. We can see that deciduous species had a huge change in that slow mechanism associated with xanthophyll pigment pool sizes, and also a pretty drastic change in the slope or the capacity for that fast photoprotective mechanism. Whereas evergreens, they show a lot more subtle changes, okay? And these plots, these are difference plots. So I subtracted PRI-0 values and ΔPRI values from mid-season to late season to visualize the degree of change. And we can clearly see that for deciduous species, except our Tilia americana here, had a greater amount of change in their slow mechanism and how they utilize that slow mechanism, where evergreens are just shows very subtle changes, okay? And for our facultative response or our fast photoprotective mechanism represented by ΔPRI, there was a huge change in our deciduous species, and actually in evergreens we see an increase in that fast response in evergreens, okay? So, summary, deciduous and evergreen species have contrasting PRI-0 and ΔPRI values at mid-growing season, and these are reflecting differences in both their fast and slow photoprotective mechanisms. And at late seasonal time points, these PRI values shifted more dramatically for deciduous species than evergreen species. And for deciduous species undergoing leaf senescence than evergreens that retain their leaves. So these results indicate that deciduous species and evergreen species have distinct photoprotective strategies that appear to be coordinated with their differences in leaf lifespan. So when we assess this, the functional trade-offs and the coordination with photoprotection, as I indicated earlier, deciduous species, these are contrasting ecological strategies that have they represent kind of the extreme examples of trade-offs in maximum photosynthesis and leaf lifespan, okay? So these are kind of the results or how they utilize these different photoprotective mechanisms. And the larger capacity for fast photoprotection kind of makes sense in this functional type because they have higher rates of photosynthesis and require that fast mechanism to regulate their light use efficiency over a larger capacity or over kind of a larger capacity, and require less investment in slow photoprotection because of that. Whereas our evergreens, they have a smaller capacity for fast photoprotection because they don't necessarily, presumably, require it because they already have constrained lower photosynthetic rates. Now, their larger investment in slow photoprotection agrees with their longer leaf lifespan. Because evergreens retain their needles, it makes sense that they need a greater investment in those pigment pools. And also, that's why we see a lower change in each of these photoprotection among these species. And while we see a greater change in deciduous species because they are shorter-lived leaves. And then kind of getting back to my field sampling that I didn't discuss much because it's complex and I don't want to be all over the place. The field PRI responses, so the sampling in shade, lower radiance positions of canopies for my deciduous and evergreen species, indicated by the stripes here. So these are lower radiance and higher radiance canopy positions. These actually, and I'm still kind of working through the analysis, these appear to agree with the differences in the PRI intercepts and slopes that we're seeing in these remote sensing studies, suggesting that our ability to derive these intercepts and slopes, representing these two distinct photoprotective mechanisms, we're able to actually do this with remote sensing platforms from airborne hyperspectral remote sensing imagery. Okay, so this is great because it suggests that we can actually measure these two different things within diverse systems, okay, without the need for necessarily proximal remote sensing or that leaf level sampling. So we can quantify all of these different responses of many different functionally diverse systems and also, improve our interpretation of PRI and the mechanisms associated with it and how that influences or is related to changes in light use efficiency and photosynthesis. And because we have identified these distinct photoprotective strategies, it can improve our interpretation of PRI. Specifically, the seasonal variation that occurred among these groups can serve as a baseline for understanding how PRI variation is associated with environmental stress. So anything that kind of deviates from those patterns could be an indicator of changes in photosynthesis. And also, this can enable us to partition PRI responses associated with kind of the predominant mechanism used by each of these functional groups. So because we see, for example, because we see a kind of deciduous species having a greater capacity for that fast response or that fast mechanism of photoprotection, we can presumably assume that that's what PRI is associated with. So yeah, that's my presentation. Thank you, everybody.
And we have some time for questions, if anyone has any.
I have one question.
Yeah, go for it.
Great talk. Yeah, so I'm I'm interested about how you define this photoprotective functioning. How to what this protection means.
Sure.
And one more question is which sensor you use, which equipment sensor you use for the PRI measurement?
Yeah. So. Right. I'm thinking of the best slide to bring up here to discuss these. Okay. So your first question, how is PRI associated with photosynthesis? Is that correct?
Photoprotection.
Photoprotection.
Yeah,
Thank you.
Yeah.
So photoprotection through the xanthophyll cycle, you see variation in PRI over short timescales are largely associated with the operation of the xanthophyll cycle. So when environmental conditions are changing dynamically, plants adjust their light use efficiency through this mechanism, and it kind of fluctuates and allows them to adjust their photosynthesis over short timescales. Whereas we see constitutive changes in those xanthophyll pigment pool sizes are associated with long-term changes in photosynthetic activity. So we see this, for example, when plants are exposed to chronic stress due to seasonal changes in temperature. We also can see this constitutive change. Evergreens is a great example because they actually increase the amount of xanthophyll pigments, this constitutive investment in those pigments, and it's associated with deep photosynthetic downregulation during the winter. So it allows, since they don't drop their needles, it allows them to dissipate all of that excess light energy off and maintaining deep photosynthetic downregulation. So they represent kind of distinct mechanisms that operate on contrasting timescales. So this is kind of the short-term quick modulation of photosynthesis, while this is kind of a larger kind of longer timescale operation of photoprotection.
Okay.
Yeah, and then your second question about the instrument that I'm using, are you familiar with PP systems, unispec? It's a multi-spec. Yeah,
I don't know.
Yeah,
I don't know. I'm just curious.
it's a multispectral instrument. It covers 350 nanometers to about like a little over 1,000 nanometers. With a bandwidth of like 2 points. Yeah, yeah, nanometers.
Okay.
Some in between 2 or 3. It's fine, and I mean, it's perfect. Well, this instrument it is perfect for this proximal leaf level sampling, just because of the internal halogen source that allows me to quickly expose dark adapted leaves and monitor these kinetic changes in that xanthophyll response.
Okay.
Yeah, absolutely. What's up?
Are there, I guess, so if I understand from the discussion or summary slides correctly, there's a smaller capacity for photoprotection in evergreens, so like a slower response. Is there implications for like climate change, how that would affect evergreens versus deciduous trees, or is that kind of separate? I don't know.
Sure. That's a great question. I think that gets, as much as I think people have focused on like functional traits and functional groups and how they acquire and utilize resources differently and what that means for their tolerance to environmental stress or changes in environmental conditions. I think viewing this through photoprotection and different photoprotective strategies, will enable us to potentially detect, photoprotective strategies that are successful under particular changes in environmental conditions. So, maybe that would be an avenue in understanding, what mechanisms are best suited for certain conditions. And then that can potentially help us predict, what that means for changes in functional diversity or plant diversity within that ecosystem, and then implications for ecosystem function. What's up?
So I'm kind of curious about the upscaling on this from a handheld sensor to some kind of remote sensing.
Yeah,
So when you're measuring PRI, you're presumably measuring some kind of reflectance, backscattering from the leaf, right?
absolutely.
And so if you're doing that for an individual leaf, the conditions are going to be relatively homogeneous over a given period of time, right? But if you're like doing this for a forest canopy where you have like large gradients and light availability,
Yeah.
water stress, nutrients, but also assuming that like you're comparing two remotely sensed images, right? Like you're not measuring the same leaf at
Yeah,
the time, right? Do
absolutely.
you think that this PRI index is like sensitive enough to actually capture that change, or are you just like measuring environmental variation and then like misdetecting that as like, plant functional response to something? Like, how do you make sure that you're actually capturing the plant responding to something as opposed to variation in space and time?
I see. Okay, so you're asking, how do I know whether or not PRI is actually associated with these underlying mechanisms, or?
Yeah, as opposed to like just variation in, some kind of reflectance of something.
I see. So like potentially like soil effects, and is that what you're
Or just
suggesting?
variation in like within a canopy, right? Or the leaves are probably going to be affected by light or like reflecting light differently depending on.
Yeah, okay. That's a great question. So this kind of gets, and I've been having trouble with this as well, because there's tons and tons of literature that's comparing sun and shade leaves. And everybody has shown, there's, a lot of evidence showing that differences in canopy light gradients result in variation, not only in the amount of xanthophyll cycle activity that's occurring, but also these differences in those intercepts or those pigment pool variation. What's interesting though is, and I wish I could have gotten my analysis done to include it in my presentation, is actually the shade, my differences in my sampling PRI from shade and sun actually appear to be fair estimates and fair correlation with these PRI slopes and intercepts between our kind of contrasting functional groups here. But I think that's a very good question, especially when you're comparing these differences in deciduous and evergreen. They have completely different canopy structure and leaf structure, and therefore they also have different like light environments. Their light gradients are also going to change. So I'm very curious in how that's kind of affecting these PRI responses. But from what I can tell, and kind of based on a lot of existing literature looking at this, is that they kind of match up. Regardless of all these underlying mechanisms that are going on, I think there's actually is a fair estimate. And that's kind of what I'm hoping to do next in my research, is actually look at the scaling from leaf to the canopy level. Yeah.
I guess my question kind of goes along with that. So this is a great chart, and when understanding that light gradient affect that occurs, and you have that PRI delta value. So in your work, do you actually attenuate when you're collecting across that gradient? Because inherently, even just a 1 foot difference in your sampling would throw off a PRI value by a small percentage, but a percentage nonetheless.
Yeah.
Not only that, one of my other questions is when you're like from the cornfields and that stuff, it's a little bit different, where we can actually calculate when is peak senescence or relative senescence, peak GPP values, and we could attenuate values in terms of understanding light use efficiency accordingly. Do you do the same when you're sampling on understanding what is that senescence value and when is peak senescence, and then you would go out and sample as well? Because your delta PRI values, even a 5-day change would temporally would change that delta value
Yeah.
because photosynthetically, what's occurring is fundamentally different in a senescence period.
Right. Okay, so the first question you had asked was talking about you were getting at the effects of canopy variation and how that's going to be influencing these different kind of responses, and then what that means for our ability to relate this then back to light use efficiency.
Well, just the value you're calculating itself, right? Is there any attenuation towards, the leaf height or, compared to the
Yeah.
PRI value?
Yeah,
That's the simplest way to put that.
absolutely. I collected irradiance from each one of the canopy positions that I'd sampled. So I sampled at 2 positions on kind of north and south cardinal directions of the canopy. And this was like the most difficult part of the sampling, it's because light is so critical to driving variation in my PRI responses, that I not only had to, sample, this is kind of a schematic of a sampling pole with a little quantum sensor on top of it, but I had to, ensure that my light conditions here are also similar to where I was doing reflectance sampling. So I ensured that those conditions matched up as a way of preserving that PRI response that we would see in the canopy from airborne observations. But you bring up a really interesting point, because I've been trying to work with this, like, okay, so if we have differences not only in like the the intercepts representing differences in those xanthophyll pool sizes between sun and shade, and then also presumably differences in the amount of xanthophyll cycle activity that's going on, how do we parse that apart? How can we actually derive like an estimate of a canopy slope and intercept? And that's kind of what my research is getting to now, is using this leaf level data, and how can we parse responses between the two, and how does that scale with our remote sensing data as well. And I did collect SIF, leaf level SIF data. I haven't gotten to it yet, so hopefully that will provide a little bit more insight on kind of what these mean for light use efficiency. But I also do plan on doing more sampling, kind of increasing the number of canopies that I sample, as well as a faster, including kind of maybe an instrument that will allow me to get an estimate of light use efficiency, like a Lycor 600 that I can just get a quick little estimate of. Anyway. Who's up? Yeah.
Thank you so much for a great presentation. I've been struggling to understand the significance of the study. How does it help us to understand the climate change? How does it help? Because we know evergreen, which therefore it's green for the whole season, and the deciduous plant that you took their short period. That one we know. So you've explained the mechanism and we've seen the graph. So how will it help us? In order to mitigate the climate change, how to extend, I don't know. Okay, you try to explain, maybe you can improve more on how to, for us like layman, because I don't know the mechanism, xanthophyll, people outside the academia won't understand that.
Yeah.
But it should be better to find out how will they understand the importance of what you are doing. That's one. Secondly, I just wanted to know if the height of the trees can have any impact on API, height, age, size of the leaf. You think that wider leaves, will reflect the light the same at the same pace as the small leaf, especially in your case study? I don't know, because you know much. Will you tell us the role, the impact of the age of the tree this the height, if I have a shorter tree, a small tree like this and a large tree like this, will they reflect light at the same rate?
Sure.
Yep.
What's nice about my study is I completely and totally avoid all those canopy effects because I'm sampling at the leaf level. And I do think you're right. I mean, I think our understanding of how photoprotection varies across different species is still, relatively new and kind of young in the field of ecology. All of those has an effect, and not only just kind of at the leaf level, but also the branch, how the leaves are oriented and clumped together within the canopy. It's kind of what I think is so fascinating about remote sensing is it's kind of this unified expression of all these different kind of across the biological scale within an individual. And all of that influences kind of the responses of reflected light that we get. So that's a really interesting question, and how kind of the variation in each one of those factors that you had suggested, how does that influence our ability to measure plant stress or changes in photosynthesis? So I think that, yeah, that's a great question. And it's something I've been thinking about as well. And your first comment was my inability to or not inability, but how do I relate this to people that aren't in this field is, so PRI is an indicator of plant stress, and it is largely associated with changes in photosynthetic activity. But the problem is it's influenced by those two different distinct mechanisms, right? That are changing photosynthesis over short and long timescales. And that's difficult when we're trying to interpret PRI. What does this mean? What mechanisms are associated with whatever we're measuring here, okay? And by kind of identifying that these species have contrasting photoprotective strategies, if I'm able to identify in, let's say, a stand, that there's a group of these are deciduous species, based on their photoprotective strategy, I know potentially, presumably, hopefully, that PRI is largely going to be associated with that fast photoprotective mechanism. Whereas variation in our evergreens are probably going to be largely that pigment poolside effect. But those represent different mechanisms and have different implications for changes in photosynthesis, and I'm still trying to work that out, what that exactly means. But those are great questions. These are kind of the underlying ecological questions that I'm wondering. What does all this mean? How can it inform us about changes in ecosystem function, or kind of be a pre-visual indicator of changes in composition? So those are great questions. Did that answer?
Yeah, for me, I understand well because I took the class that Professor Gamon is teaching.
Oh, cool.
So I relate it with the climate change because what you are doing is a major indicator of climate change.
Mm-hmm.
The greening, for instance, there's been paper published about in northern part of America there is greening observed. So we can track the indicator that you are doing or the mechanism that you've shown us here in order to know what is going on in the northern part of America compared to Africa.
Sure.
So you see, so that indicator is very important. So what you are doing, I can say, is very important to even to detect signs, early signs of climate change.
Absolutely.
Yeah, even the effect of the climate change. Extended, we see greening all the time to some part of the world, but other part we don't see. What's going on?
Sure.
That's the mechanism underlying, part of the mechanism underlying what we observe.
Absolutely. Yeah. Cool. And what's so neat about this, and what's so that there's so much potential is, we don't have, there's many studies have assumed that PRI variation is just the operation of the xanthophyll cycle. And nobody really actually looks at changes in these constitutive pigment pool sizes and what that means for stress or a stress response. So, and kind of the time scale coordination between both of these responses is unknown. So part of what I'm interested in is how does changes or variation in this slope or this fast photoprotective mechanism, how does that affect the timescale operation of these kind of long-term changes in photoprotection or photosynthetic activity that, and that's largely unknown. What's up?
Okay, so I want to add on what you said about the question. So also PRI is a great input for the light use efficiency model, which is good in predicting the GPP. Also, like expanding the PRI from Deciduous to Evergreen is a great research because the new spectral satellite pace can help us to use light efficiency model over all the globe. So expanding the PRI from Deciduous to Evergreen can help us to expand that light use efficiency model so we can track the GPP trends all over the globe. Yeah.
No, a question?
Yeah, it's kind of been answered. I guess I was gonna kind of ask about like larger applications of this. Like, would you expect to see a difference in the PRI between like kind of the leaves you sampled, which are like a singular tree on campus, like getting tons of light, or then like a tree like in a forest, and then maybe broader applications, like could you see maybe the stress response of like there's been some deforestation, or maybe just phenotypic plasticity even between different forests with the same species and their response to different light, which this has all kind of been hit on, but.
No, this is a great question. So I think what's so cool about identifying these kind of contrasting photoprotective strategies is we can use these responses, these seasonal patterns of variation between our two functional groups, and that'll serve as like a baseline. And then anything kind of that deviates from that baseline potentially is an indicator of, well, these plants are under stress. And we can refer then to these seasonal patterns. Anything that deviates from that might be an indicator of they're experiencing different environmental conditions, or they have been like predisposed to environmental conditions based on just variation across biomes and environmental conditions and stuff like that. What else is that?
Yeah, that's answers it. Thank you.
We would have time for one more question.
Are you going to be looking at this as a function of environmental gradients in the future? Because like if you have these strong seasonal patterns, they might mask the ability of PRI to indicate stress potentially. But like, are you conducting the same experiment just with stressed versus unstressed leaves across like some kind of spectrum, if it's nutrients or water, would be super.
That's what that was my original plan that I wanted to do. Out at Horning Forest, they have kind of a rows of relatively young trees that are a lot more easier to access their canopies. But I mean, it's just a lot of work setting up treatments. I would very much like to do that. I can't do that to the trees on campus here. Otherwise the groundskeepers would be very angry with me.
Do you use like climate gradient across Nebraska?
I suppose so. Yeah, absolutely. You had said, these seasonal patterns might mask our ability to assess kind of fine scale changes in photosynthesis. And this kind of gets back to Serge was saying, is PRI scales well with GPP? Well, because I think these constitutive changes are largely, the major or main mechanism driving seasonal variation in GPP. So across a lot of studies, we see, okay, PRI is a great estimator of GPP. Yeah, because we're largely just kind of estimating it to these constitutive changes rather than kind of this fine scale dynamic changes that we see associated with the xanthophyll cycle. So kind of analogous to your question is, or I think what's interesting about your question is how much more does this, our ability to kind of measure this and quantify this fast response, how well does that inform us of these like major changes, seasonal changes in photosynthetic activity? Does that make sense?
Yeah, I mean, I guess one like criticism of that might be that you're conflating time and spatial scales, because I'm assuming GPP is measured over the growing period, and then you have like an average GPP value per
Sure.
aerial extent, and that is going to reflect the PRI intercept, because that is related to the nutrient content of the leaf or the chlorophyll content, right?
Sure.
And I don't know if like a short-term adjustment in that xanthophyll cycle would be a strong predictor of, seasonally average GPP, like, because those are operating on such different timescales.
Sure, but that's what's so interesting, and I think is a lot of potential for this, is how do the timescale operations of both of these complement each other? So can this serve as a pre-visual indicator of changes associated with that? And that's could be due to, if we, maybe let's say there's a drought event and we see kind of a high response associated with this, followed by like changes in leaf phenology, or altered phenological patterns. Maybe this could serve as a pre-visual indicator of those large-term or long-term changes in GPP. But I understand what you're saying. Yeah, yeah, that's a good question. Okay.
All right, thank you, Alec.
Yeah, I can. Great thing you're reminding you. You were in the morning, right?
Yeah.
Aren't you?